Okay, so we're going to dive into something really interesting today. It's about this tiny, almost invisible organism that costs the global poultry industry millions of dollars. It all starts with a simple sneeze, but the impact, well, it's absolutely staggering. Let's talk about mycoplasma. All right, let's just start with a number. 75 cents. Now you might think, 75 cents, what's that? Well, that's the estimated loss for every single bird that gets infected with mycoplasma. Now think about it. Multiply that by the millions and millions of chickens in production all over the world. All of a sudden, that little sneeze in the henhouse turns into a massive, multi-million-dollar global problem.
So that really brings us to the big question, right? How can this microscopic germ, one of the smallest bacteria known to science, cause such a huge financial drain on an entire industry? To figure that out, we've got to follow the money.
Okay, first up, let's really break down where these losses are coming from. Because it's not just one thing. It's more like a cascade of problems that hits pretty much every single stage of production.
And what's so wild is just how the damage stacks up. So for meat birds, you're looking at poorer growth and, get this, up to 15% more condemnations at the processing plant. For the egg layers, it can literally steal between 8 and 15 eggs from every single hen. But wait, the costs don't even stop there. You've got lower hatch rates, you're spending all this money on medication, and maybe the biggest problem of all is that mycoplasma weakens the birds. That makes them vulnerable to a whole bunch of other diseases. The costs, they just spiral out of control.
So what exactly is this tiny little culprit? To really get why it's so destructive, we have to look at its biology. Because its most unique feature, well, that's also its greatest weapon.
So mycoplasma are the smallest bacteria out there that can actually survive on their own. But the really crucial part, the thing you have to remember, is right there at the end. They lack a cell wall. Most bacteria have this rigid outer wall, right? Mycoplasma doesn't. It's basically just a flimsy, flexible membrane holding all its guts in. And that one little detail, it changes absolutely everything.
Here's a good way to think about it. A typical bacterium is like a knight in a full suit of rigid armor. A lot of our most common antibiotics, like penicillin, work by attacking that armor, the cell wall. You break the armor, the bacterium falls apart. But mycoplasma, it's totally different. It's more like a spy. There's no armor to attack. All it has is that flexible membrane, which means all those antibiotics that target cell walls are, well, they're completely useless against it.
This no-wall thing gives mycoplasma a bunch of advantages, almost like superpowers. First, it's stealthy. It can literally wear the host's own proteins as a disguise to hide from the immune system. Second, it's resistant. Like we just said, it's naturally immune to a whole class of antibiotics. And finally, it's invasive. It doesn't just hang around on the outside, it can actually invade the bird's own cells. That creates these chronic, persistent infections that are just incredibly hard to get rid of.
Okay, so if you've got this stealthy, persistent enemy, how do you even know it's there in the first place? And that brings us to the detective work, diagnosis. For a long time, the gold standard was to actually grow the bacteria in a lab. You take a swab from a sick bird, put it in this really complex special goo, and then you wait. And you really wait. It can take 3 to 5 days, sometimes even longer, just to see if anything grows at all. Sure, it's reliable, but man, it is not fast. And this is where you see this huge leap forward in diagnostics. Culturing is still super important for some things, but PCR technology has been a total game changer. Instead of waiting days and days for bacteria to grow, PCR can find the bacterial DNA, and it doesn't matter if it's live or dead, in just a few hours. And that speed, that is absolutely critical when you're trying to stop an outbreak in its tracks.
All right, so you found the enemy. Now what? How do you fight it? Well, let's look at the main strategies for the counteroffensive, using vaccines to prevent it and antibiotics to treat it.
Now, when you're talking about vaccines, it's all about making strategic trade-offs. On one hand, you've got the F-strain vaccine. It's super protective and can even push the more dangerous wild strains right out of a flock. But here's the catch, it can actually make turkeys and young meat chickens sick. So on the other hand, you've got safer options like TS11 and 6/85. They're much less likely to cause any problems or spread around, but they just don't offer that same powerhouse level of protection you get from the F-strain. It's a constant balancing act for the vets on the ground.
So what about actually treating sick birds? Well, since drugs like penicillin are totally useless, vets have to turn to a completely different arsenal. The big guns here are classes of drugs like tetracyclines and macrolides. See, these antibiotics don't even bother looking for a cell wall. They go straight for the bacteria's internal machinery,
shutting down its ability to make proteins and basically stopping it cold.
But, and this is a big but, using these specific antibiotics on the farm brings up one final and really important complication. And this is one that goes way beyond the henhouse.
So here's the problem. It's like we're all dipping into a shared medicine cabinet. A lot of the antibiotics that work on mycoplasma in chickens, we're talking about things like tetracyclines and fluoroquinolones, well, those are also critically important for treating infections in us, in people. And that overlap, that creates a huge potential problem.
Okay, so here's the real risk. Every single time we use these antibiotics in animals, we're creating what's called selective pressure. Basically, we kill off all the weak bacteria, but we leave the strong ones, the ones that happen to have resistance genes, to survive and multiply. And the big fear is that this resistance can spread, turning farms into these reservoirs for superbugs. That could make our own medicines way less effective right when we need them the most.
And listen, this is not just some theoretical idea. The source material says it directly, and I'm quoting here. The use of antimicrobial agents in food-producing animals has become an important public health issue due to the spread of microbial resistance, which might possibly be transferred to human pathogens. I mean, that is a pretty stark warning about how connected animal health and human health really are.
So this really leaves us with a huge dilemma, doesn't it? We have to control diseases like mycoplasmosis to make sure we have a stable food supply. But how do we protect our animals without giving up the power of our own life-saving medicines for the future? It's a tough question that sits right at the intersection of our dinner plate and the doctor's office. And figuring out how to strike that balance, that's going to define the future for both.